consequences. Pesticide contamination of the agriculture soil poses an escalating threat to non-target invertebrates, with earthworms serving as a vital sentinels of soil ecosystem. Tolfenpyrite induced oxidative stress and the histopathological disruption, deeper ecological imbalance with far-reaching the consequences of the food safety and mainly soil biodiversity. Bridging the ecotoxicology with the One Health paradigm, this study underscores the indispensable role of veterinary and environmental science in safeguarding the integrity of our food system. So the tolfenpyrite is a pyrosol group of pesticide which is widely used in the southern part of India, especially in the Kerala. Since it is soluble in water, many of the farmers tends to use it in an indiscriminate manner. So the objective of this study is to find the LC50 of the tolfenpyrite and to evaluate the subacute effects of the tolfenpyrite and to assess the dose-dependent toxicity. So next comes the materials and method. The experiment was conducted based on the guidelines suggested by the OECD 207. So the artificial soil method was used, and the earthworm is Eutrellus eugenia, a species of an earthworm. It is also called as African night crawler, and the pesticide used was tolfenpyrite. To find out the LC50, the number of days were 14 days, and for the each dose we had 3 replicates. In 3 replicates there will be a 12 number of earthworms, and based on that we derived the LC50, and then the subacute toxicity study was conducted in which 4 replicates, and based on that each replicate there will be 12 earthworms, and the number of days of study was 28 days. The parameter assessed were morphology and behavior abnormality and histopathological antioxidant parameters. And finally, statistical analysis was done using the GraphPad Prism 8.2. So based on the 0 mortality and the 100 mortality, LC50 was derived as 120 mg per kg. So next comes the results of the subacute study. First, the histopathology. Histopathology control 1/40th of the LC50, 1/20th of the LC50, 1/10th of the LC50. There were no much difference between the control and the vehicle control of the subacute groups. And A denotes the marked cellular degeneration, which includes the epidermal and the muscular layer. Whereas the B, which is denotes as the goblet cell hyperplasia, which is a reason for the excess of the mucus promotions. And E, which denotes the degeneration of the chlororgan tissue, which is the earthworm doesn't have the liver, which the chlororgan tissue acts as a liver and it will filter the toxicity. So this degeneration results in the toxicity of the tolfenpyrite. F, the reduced spermatic follicles. The arrow mark denotes the spermatic follicles and in which the sperms will be present. So when compared to the control group, 1/40th, 1/20th and 1/10th, there was a drastic difference in the spermatic follicle reduction. And the G graph and the G image denotes the longitudinal section which correlates with the cross sections of the earthworms. The morphological alterations include segmental coiling, excessive mucus production, which can be correlated with the goblet cell hyperplasia, bulging, clitellar shrinkage, body coiling, body bleeding, and spasticity, which is an indicative of the neurotransmitter. And next comes the antioxidant parameters. There were a decrease in the production of MDA, which caused the activity of lipid peroxidase to decrease, and there were a statistically significant increase in all catalase, SOD, acetylcholinesterase, GPX, and GSH, which all indicates that the toxicity were dose-dependent manner, and moreover there were a significant difference between the high dose and the moderate dose, which states that the toxicity is more in the high group. And last, the conclusion: veterinarians are the essential to the modern agriculture, not only as the animal doctors, but as the food safety experts, public health protectors, agriculture advisor, and environmental stewards. Their role directly supports the sustainable farming, rural economics, and global food security. And thank you. This is my conclusion.
Now the topic is open for discussion.